What if you could take someone who is on their deathbed and find some way to hibernate them until the sort of critical cure for the disease comes online?
The ability to freeze time for humans. I didn't actually think that was something you could go work on, so apparently it is.
Our long-term goal is reversible whole body cryopreservation for medical hibernation. But in the near term, what we work on is reversibly cryopreserving single human organs to help transplant patients get organs more efficiently. Making time not a variable changes the whole paradigm. One thing I love about the field of cryopreservation is I think, the problem speaks for itself. Water expands when it forms ice. That's just hard for your tissue to take without substantial damage. And the cool thing is that there's sort of a temperature below which ice formation stops happening. So basically, if you can traverse and you can get below that without ice formation, then you're good. We already reversibly cryopreserve tissue, including human tissue, all the time. And we do it for very long time periods. There are kids who were literally cryopreserved for 30 years as tiny embryos. And so the main question is not, is this possible to do at all? It's, is it possible to scale up?
Given that's true, why don't you think it's been worked on?
Hi, listeners. Welcome back to No Priors. Today, I'm really excited to be here with Laura Deming, previously the founder of the Longevity Fund, and now the co-founder and CEO of Until. We're going to talk about how Until is progressing the frontier of reversible cryopreservation, or freezing living things and waking them back up, beginning with human organs, progressing to small animals, and hopefully making progress on the whole body. It sounds like science fiction, but we'll talk about some of the scientific challenges, where we are today, and the implications if this is possible. Thanks so much. Welcome, Laura. Laura, thanks so much for doing this.
Yeah, thanks for having me.
I've been so looking forward to this since our Pantheon Watch sessions. We're talking about upload and the nature of consciousness. But one thing that you don't know is that my very long ago wished for technologies that I wanted to exist were telepathy and upload, and the ability to freeze time for humans. I didn't actually think that was something you could go work on. So apparently it is. How do you end up working on that or being interested in longevity at all?
There's two different questions. So yeah, I come from a longevity background, but in my mind, reversible cryopreservation is applicable a bit outside of that as well. I don't know. I mean, I think I'm really obsessed with areas that feel like they should be worked on but aren't. And when I was a kid, I think naively, just growing up, that seemed really obvious for longevity. And it's really surprising to realize that it's not the case that most people are working on that explicitly as a goal. And in fact, that it's kind of I think longevity and aging kind of occupy this weird realm where, because they're not explicitly diseases in a way that's fully socially recognized yet, they're not seen as valid to work on. But that's not really for, I think, technical reasons on some level. It's more for classification reasons, because you can extend the lifespan of sort of many different organisms using technology. And how much you can do that in humans, we have no idea. And it could be very small technology, but just sort of I think longevity is interesting because it feels like an area where there's a social blind spot around something, and I find those very interesting.
Perhaps I was just, I'm sure this is true, not very observant as a school-aged child, but I don't think I even understood aging was a concept that I should consider at all. And so how did you end up thinking about it in any depth?
I grew up in a pretty odd setup. So, I was in New Zealand, I was homeschooled. I didn't really have I didn't go to a normal biology class. I was kind of, by myself in the house.
I imagine you staring at a field of sheep and then being someday we're going to get old. I should do something about this.
Yeah, no, that would have been the farm that we had for a little bit. But I remember one thing that really stood out to me was at some point when I was a kid, I was thinking about how long people in my life were going to live and how old they were. And for some reason, it made a lot of sense to me that everyone should live until they were 10 years old and then die immediately at 10 years old. That seemed like some hypothesis. I didn't really know how old people were. And so working backwards, I was like, oh, my dad must be maybe 8 and my mom's younger than him, so maybe 7. But I think one thing that was really striking was realizing that we don't all live until a certain age and then we die. In fact, we don't, know what determines how long we live. That was very interesting, right? This idea that almost if we all lived until 10 years old and then we died immediately at 10, I would feel much more confident with the idea that longevity is some kind of inmalleable, hard limit. But the idea that there was uncertainty about that, it was just really interesting. And then sort of what are the factors behind that uncertainty?
So I'm going to fast forward through a bunch of lab work you did and going to MIT and being a Thiel Fellow. Why a longevity fund?
Me just being very literal, at the time that I was interested in longevity, I think if you ask the average person in the field, what's the big problem? Most people would say, well, we just can't get enough funding for our projects. And so that's the big problem. And I just took that literally, like when I was a teenager. So
I'll solve
went
that
out.
problem.
Yeah, I was just like literally like I should just get a lot of money to like help push longevity drugs forward. And the name for that happened to be venture capital fund, but it definitely wasn't working downstream of the idea of venture capital. Like that came after this idea of like just getting money for projects that should have money and didn't.
You did this for a number of years. What triggered the like sort of change in how you were going to spend your attention to cryopreservation?
I think just like cryopreservation is one of the coolest, like I think it's not like to pardon the pun, but I think it's one of the coolest, most interesting, like best problems ever. Like I think like, I mean, if you, from so many different angles, like I think if you're obsessed with counterfactual impact, like if you're obsessed with like just technical delight, like just raw sheer like technical interest and diversity and just like the lot of technical parts of the problem. And then also I think from perspective of social impact, like it's such an interesting problem from like how it's perceived and then like what are the different factors of that. It was like 0 to 1. It was like I remember just seeing the problem clearly for the first time. I mean, a lot of the people in my life, I think, had been aware of it, but it took me so long to really, I think, see it clearly for myself. But then it was just like 0 to 1 of like, this is the only thing that I could imagine pouring the next decade of like my work into. That was after I kind of done kind of the first set of work with the fund. I was kind of like thinking like, what is the next like big thing?
With your co-founder, Hunter, was it like an immediately obvious, yes, we should work on this together?
He's one of the few people where if I ask him a question, like he'll come back the next day and give me an answer where he's literally thought through from first principles, like what the correct answer is. Like I remember at some point, I realized that he'd written a doc on like the principles of like, cryopreservation. Like most people, like would write that from the literature or kind of like citing different sort of sources at various levels of granularity. Hunter went back and like re-derived fundamental laws of stat mech as part of like, this is what you should know in cryopreservation doc. Like I really admire how much he builds up from like really simple models to try to create coherent like technical pictures that are more complex. He's like the most fun, interesting, best person to work with ever, for sure.
And it wasn't a hard sell, like we should go work on this problem in particular.
It was not a hard sell, but it was not an interesting way. So I think one thing that I like love about the field of cryopreservation is I think it's very compelling. Like if you like it's one of those things where if you like the problem speaks for itself. And so I remember telling Hunter about it in our first call, and he basically was like, oh, I don't buy it. He told me later that he didn't tell me in the call. But then he went off and thought about it for a couple of days and like really thought about like what we knew about ice formation, like did, some like basic back of the envelope math and came back and he was like, oh, wait, this seems like actually feasible, or like this seems like it's in the realm of possibility in a way that is very different from my initial intuition. And that sort of like conjugation is just so interesting to me.
Maybe it's useful to zoom out and just say, like, what is the goal of Until?
I would think about our goal as trying to create a new form of critical care. The example that I would give that is sort of the core of the company is there's some years where certain diseases such as like metastatic melanoma go from being like sort of in a single year, like metastatic melanoma went from being something that you had like a 6 to 9 month prognosis, like less than a year of expected survival, to with like, new combination immunotherapies, you might have a decade plus of expected survival, or like 50% of people sort of surviving over a decade, without getting sort of death from melanoma. In fact, they were starting to die of other things at that point. The tagline is like, single years can make the difference between a patient dying of terminal illness and like living long enough to make the critical cure. But right now, like there's no way to press pause on their biological time. So like, what if you had an ambulance to the future, right? Like, what if you could take someone who is on their deathbed and, find some way to, just sort of hibernate them basically until the sort of critical cure for the disease comes online. And like, and in this context, we're not talking about necessarily decades or kind of like, much longer than that. Initially, it's just kind of in the context of like, when there's a window where you could imagine like a critical trials being done for a drug that, were they eligible for it, could make a huge difference for their disease. To give an example of like the need too, it's like my sort of co-founder's father-in-law had this happen to him in the sense that he got sort of advanced cancer that would have been treatable or addressable by a therapy that came out basically a couple months after he was no longer eligible for the therapy. And he like missed, the critical clinical trial by like, a couple months. And so it's sort of like that level of urgency that like someone in that position shouldn't have to, miss a critical therapy because there's a couple month difference in when they got their sort of disease and when the therapy became available.
And from a just like product perspective, that means you need to do whole body cryopreservation.
Yeah. So like to give context on technically how we think about this problem. So our long-term goal is reversible whole body cryopreservation for, medical sort of hibernation. But in the near term, what we work on is reversibly cryopreserving single human organs to help transplant patients get organs more efficiently.
I want to come back to like all of the technical challenges here and where we are and what you think the next milestones are. But because you describe it in the context of medical use, like I'm going to be honest, as a kid, I was like, well, I want to be able to freeze time because I want to be able to go to Mars too. Or you said because you've worked on longevity with this perspective, like aging is, it could be considered a disease, a health state we should work on that is credible medical science. So how do you think about those other use cases?
So an interesting thing when you start to think about like actually applying this technology is sort of what's the experience of the person, not just kind of technically for the disease, but socially. So like one of the number one reasons most people wouldn't do medical hibernation, and especially that it wouldn't be like for the most part a recreational thing, is that I think a lot of people view themselves in part defined by their social context. So like
when you say recreational thing, you mean because like you think of that as going to Mars as a recreational use case?
No, sorry. I think this case, what I'm thinking of is, some people might imagine that they would love to skip into the future just to see what happens, and maybe, like have their same number of years of life, but like be future shifted by some amount of time.
Yes.
But the number one reason that most people wouldn't do that and are also just, very wary of the idea of hibernation for themselves is,
I can't take everybody with me.
Yeah, exactly. It's this idea that like you kind of define yourself by the people around you. And so I think like, those kinds of use cases like going to Mars, like those are all kind of things that could happen and could happen with or without or like sort of could happen, will require hibernation technology for certain like definitions of mind. But I think that the thing that a lot of people will face is just like this question of like, is it worth, traveling so far to give up my current social context? And that will put a limit on like how much people want to use this for. Like, I think there's like a real cost that kind of you incur. And so it's, in my mind, it only makes sense for like really serious use cases initially, where like you literally would die because you're kind of putting on the line like your current, like all of your current social reality and like how that will evolve without you versus like, this other thing that you might want. But I mean, a lot of people might want to go to Mars, in that context, which I think I'm a little, The cost is pretty significant.
It's very hard to know what we want, though. Like a lot of people might make that decision whether or not, their ultimate happiness is higher.
Fair.
Maybe you can just break down how you think about the challenges scientifically, right? Versus, I think, to maybe even Hunter's original reaction, like, sounds like science fiction. I don't know if you can go work on that thing.
Yeah.
Right. And so if it's, crystal formation or whatever the set of challenges and what sequence you think you should solve them.
I see. Yeah. Maybe I can just give a series of facts that I think together sort of make the problem super interesting. So one fact is that ice formation is a stochastic process. So if ice just formed unilaterally in any given material past a certain point of temperature, like, just like go from 0 to 1, it's like 100%. Ice, like that might be kind of hard to think carefully about technically, but ice sort of forms through a process of random nucleation and then extension. And this is cool because you can modulate the like sort of nucleation, the rate of nucleation and extension to then modulate the probability of ice formation. And because it's probabilistic, if you can do that well enough and you can sort of spend minimal time in the temperature range where ice can nucleate, then that gives you a shot at sort of preventing a lot of ice formation. So like the number one tagline would be like, avoid ice at all costs, or like sort of avoid as much as possible. There may be some, like, I think some people might be working on technologies to cryopreserve with some sort of like ice formation, but we're focused on regimes where you're basically trying to avoid as much ice formation as possible.
Just for the non-biologists, ice formation is bad because it breaks all the cell membranes.
Yeah, so ice formation is bad because ice expands, so water expands when it forms ice. And that's just hard for your tissue to take without substantial damage. So you want to avoid ice formation. And the cool thing is that there's sort of a temperature below which ice formation stops happening. So basically, if you can traverse, let's say, going below 0 degrees or less through to around minus 130, and you can get below that without ice formation, then you're good. And the interesting is at that point, you're good for quite a long time. So there have been human embryos that were reversibly cryopreserved for the latest record was over 30 years, and then rewarmed and sort of viably used to create pregnancy. And then, sort of like there are kids who were literally cryopreserved for 30 years as tiny embryos. And so that's the last thing, which was very surprising to me, which is that like we already reversibly cryopreserve tissue, including human tissue, including whole body human tissue at that very, very small, like sort of, in a couple hundred cells stage all the time. And we do it for very long time periods, which I honestly like would have first principles been stuck on. Like, is this possible to do at all? Is it possible to scale up to a large complex biological system that has a lot of vasculature, where you're dealing with different material properties, where you have to think a lot about like perfusion and how to sort of diffuse chemicals in and out and how to like get heat in and out quickly enough. So the idea that you could pause all molecular motion and then randomly restart it and even a cell would survive that, like from first principles to me used to seem crazy. But
like, we know that works.
Yeah, it's like, wait, we already scientists just tried it and it worked. And so now the problem is like scaling that up and doing it in a way that's compatible with like, tissue health.
Yes, I think one of the things that was most wildly surprising to me, like being in your lab a little while ago, is how much it looks like people were working on what I'd consider to be like engineering problems around. It's like, oh, how do we get something to warm quickly and safely enough versus let me go work on this therapeutic.
Yeah. So actually, I feel like there's this part of the problem that I've been trying to explain externally for a long time. Every time I try to explain it, I think it comes off as like not specific or something, but it is actually one of the core reasons why I think the problem is interesting to work on, which is that like you can trade off like engineering difficulty and biological difficulty to a non-zero degree. Like not 100%. Like you can't just use engineering to solve the problem. You absolutely have biological questions, and like those questions could come out in the negative for some use cases. So like that's not saying you can just make an engineering problem, but like you can make your life easier on the biology front by building better engineering tooling. And the fact that that's possible is a huge deal. Like that is not true for most problems in biology, and it gives you a lot of leverage on the problem. This is probably interesting to maybe only like 5% people watching this, but like I think that I'm obsessed with is just the idea that like temperature is such a beautiful conceptual tool, right? It's like temperature as an idea is something that in physics took physicists hundreds of years to come up with. It links like molecular motion to a single high level measurable parameter. And just tuning temperature like sort of tells you about almost like the relative passage of time of like molecules at the nanoscale. Like that's a highly non-trivial sort of conceptual lever to have on a problem. And in biology, one of the biggest problems is like it's really hard to find powerful conceptual levers on sort of like for like nanoscale, for manipulating like the nanoscale that like have anything approaching that degree of sort of leverage. Basically, what that gives you is like you can apply a lot of theoretical sort of a lot of theoretical toolkit used in physics to model parts of this question in ways that are actually useful. And it is just not true that you can use like equations from physics to think usefully about almost any other problem in biology. There are plenty of sections like in the context of like medical devices, but like in a context where you're talking about like changing the course of a terminal illness, which this one is interesting because like, this doesn't unilaterally change it, but like it gives you the possibility of sort of more time. I think is one of the most important things to understand about the problem. I don't yet understand how to explain it in a way that is clear, but I think it's like one of the most important things to understand about the problem.
Yeah, maybe if I think about actually applying it like just very concretely to what you are doing, like if it is challenging from a organ preservation biology perspective to have a organ rewarmed like evenly throughout, then maybe the thing to do is to like change the surface area to volume ratio of like your heating device or distribute the heat in different ways without like changing your understanding of the biology, but just with new devices and technologies that you invent from the engineering perspective. Like that actually seems like a very simple example. I realize you're implying a like more fundamental view of like why temperature is just such a interesting framework to be working on from both an engineering perspective and a biology perspective. And there's like trade-offs where you put your effort here. But I think that's something that like did not occur to me at all coming into your lab and learning more about until I was like, oh, it's actually like to some degree, much simpler problem than I understood, to the point of like, well, if you can just reduce and increase temperature in these ways that are perfect through this organ, it's going to work, right?
Yeah. So I let me restate that and then give one caveat just to make sure that I can correctly. So a way that you can talk about the trade-off between engineering and biology is that like with engineering you can modulate cooling and rewarming rate just to certain extents, and then that can then change like how much what we call a cryoprotective agent or kind of chemical that modulates ice formation you add to the system. And you want to minimize the concentration of that cryoprotective agent. So
Because
that
it
you
could
have,
be toxic. Yeah.
Exactly.
Yeah.
And so as you increase cooling and rewarming rate, aka spending therefore minimal time in kind of the danger zone of ice formation, you can correspondingly decrease the concentration of cryoprotectant that you're putting in. But if you could instantaneously cool and rewarm, then you wouldn't have to put any CP in. But that's not something that we're default assuming is feasible for a large system. So that there's still always going to be a component of biology, aka like how tissue responds, especially from a toxicity perspective, to like a new chemical agent.
Where are you now in this progression? Like, should I think of it as like there's a kidney and then it seems like quite a large jump to a small animal, but maybe it's not?
Yeah, so we work on the two in parallel. So we both work on scaling up cryopreservation and rewarming technologies to kind of human organ scale. And also in parallel, we work on sort of whole rat reversible hibernation and translating technologies over from sort of what we learn on the kidney side into the rat context, as well as doing things specifically for rat.
When you started the company, did you have a timeline in your own mind?
So I think initially I was like, we could maybe make some progress and hopefully make some good products. But like the idea of whole body cryopreservation felt like that would be really far out if that was possible. I mean, I think I definitely still wouldn't put like a near-term timeline on it, but I feel like we have a much clearer roadmap, at least to begin to get there. And the first steps seem faster than I would have imagined, if that makes sense. Like, but the big unknown to get to whole body reversible is the brain. It's unclear. Like the brain can withstand a lot of change and does withstand a lot of different types of damage or change with age, for example. But it's unclear whether like what kind of injury the brain could sustain in the context of like a whole body reversible cryopreservation protocol, and then like what level of fidelity it's possible to do. So like that to be clear is a big unknown on the neuroscience side.
How do you recruit and like lead in a company that has like, let's say, an unclear like timeline around a really big scientific goal like that, like in terms of both finding people that are the right fit, motivating them, and how do you think about urgency in that context?
I mean, I think we have a pretty clear timeline for our first product, which is like get reversibly cryopreserved So basically, transplant patients today surprisingly frequently miss organs that are en route to them because there's a timing problem. So like, organs expire very quickly after they become available from a donor.
That's very unfortunate.
Yeah. And it's crazy because it's like they're like one of the most precious resources we know of. And yet like people regularly charter private jets. Like it's like you're putting a surgeon on a private jet to go pick up an organ, get it back to a patient in time.
Yeah.
Like, and you're doing all that at the last minute and scheduling the patient for surgery at the last minute. So the patient has to wait within like a 2-hour radius of a transplant center with a pager on them or like a notification device at all times. And so the first product that we're aiming towards is just, being able to pause time for the organs. The patient doesn't, so, you can take as long as you need to get the organ to the patient. And that's very near term. Like that's not long-term ethical. That's like, we're aiming to get that into like preclinical studies and into the clinic as quickly as possible. So I think it helps to have a very concrete goal that clearly is relevant to our long-term goal, right? I think another thing that was inspiring to us about that product was like, if we're at all talking about whole body reversible cryopreservation and we can't make a dent on that problem, like
there's no version of not going through it. Yeah.
It's one of those things where like, it's like if you're serious, that should be doable. And if like you can't do that, then like you're not the company to like do the long-term thing. So that it was nice for us to like have a very clear benchmark for ourselves of like, are we correct that this is a tractable technology on that scale?
I guess like, is it a challenge or to lead beyond that? Because everybody understands the company has a broader mission, or is it just focus on step one?
I think there's the possibility that's difficult, but I think right now I feel good about sort of our ability to communicate around that, which is like, I think if we were like, it's 100% possible to do whole body reversible cryopreservation, there's no question, we're certain, like then we would just be bullshitters and then we wouldn't be able to recruit because it's, there's a lot of technical risk and there's a lot of uncertainty between here and there. But I think what we can accurately expose the models that we use to think about the problem, like why we think that this is at all like possibly in scope. And like, what we're testing to, sort of try and get closer to there. And I think the problem is interesting enough that, it's similar to Hunter, it's sort of like really good people tend to be skeptical at first because there's an intuition that it shouldn't work. But then like they'll see a lot of data very quickly where it's like, wait, I wouldn't have thought that was possible intuitively, but that seems to be possible. And so like, let me think about this from first principles.
What's another data point that you think matters besides like embryos can be frozen?
Embryos can be reversibly cryopreserved. I mean, there's work from the existing cryobiology community. So from John Bischoff's group, and then predated by that, also, Greg Fahey has done excellent work sort of looking at reversible cryopreservation in kidneys and showing like you can reversibly cryopreserve a kidney, rewarm it, put it into a rat that does not have another kidney, and that rat returns to normal function after about a month. So it's sort of like, even just on the whole mammalian organ scale, like this is not a problem that's entirely out in the wilderness. It's something that like is even academically tractable right now.
Given that's true, why don't you think it's been worked on? I realize I'm asking to answer for other people, but I'm like, well, that's very odd.
Yeah, I mean, I think even the field of like organ reversible cryopreservation really had trouble for a while attracting the resources that would be required to scale it. And I really give a lot of credit to the field pioneers. Like, Greg, is just an incredible example of somebody who fought tirelessly to make this field a reality and sort of make vitrification a thing that people were taking seriously. And, like through a long period where I think it just wasn't kind of something to focus on. But I don't know how to put it, but like in venture, like I think my whole job sort of in that part of my life is like picking trends that like sort of feel like, oh, this thing just feels weird, or it seems kind of hard to think about, but technically like there's nothing kind of that corresponds to that. And I think like, for example, like whole body reversible cryopreservation, like now seen as just much more kind of reasonable, but whole body reversible cryopreservation, definitely an area where I think there's just like so much possibility for conflation with like really extreme, like it's hard to talk about it rationally. People either like love it and they love it so much that they won't question it, or it's like they're worried that thinking about that, associating with it is a little bit too science fiction for kind of where it might be optimal for them to be focused. And so I think like I'm kind of dancing around some things that like are still a bit anti-mimetic around it, but yeah.
The study of longevity has become much more mainstream over the last 5 years or so, both academically and in terms of consumer interest, right? And I think those two things are linked. Do you hope that happens with cryopreservation?
Aging becoming something that like one of the best, like someone who's seen as one of the best like next generation professors chooses to work on without shame or fear of like not getting a grant is great. That part of aging being downstream is great. And like I definitely hope that happens for cryopreservation, specifically also for cryopreservation of like a variety of tissues, including like neural tissue, and including like things related to whole body work. Not exclusively, but like basically those topics are like seen as something that you're like it's fine to work on.
Maybe just like if you go to paint a picture of organ transplantation is transformed by, until like in what ways? Like how do you think that will change either what type of care patients can receive, or even do you think that has any impact on how people think about the speed of medical research?
I think the thing that feels the most compelling for me is just like the experience of the transplant patient. It's really, I think, constricting. It's like you're waiting for a life-changing surgery and you have no idea when it will even be scheduled, and you can't go on vacation or really leave, like, and you have to like move, you have to be like close to the place where you will get surgery, and you can't leave a certain radius of like travel distance to that place for fear of losing out on your life-changing surgery. Like that is such a
on surgery center house arrest.
Yeah. That's such a crazy proposition. And then I also think, right now for matching, it's like everything's done at literally the last minute. It's like someone dies, an organ becomes available, and like you're just kind of calling around trying to find like what patient is available to get this organ, and you don't have that much time to make the optimal match, right? It's sort of like who can get to the hospital?
Yeah.
Who's high on the wings?
Yikes.
And so I think just like one sort of transplant surgeon that we've been working with, the way he described it was just making time not a variable changes the whole paradigm. And I don't think that will happen overnight, obviously, but like I think the dream would be that like everyone in the ecosystem has the time they need to like, make the best possible decisions, sort of like do things in a way that feels the best for them. Like a surgeon doesn't have to stay up overnight the same day that they flew out to get the organ to do the surgery. They can like wait sort of until it's like the best time for them instead of doing it like literally, like as soon as they land, like they go into operation like that. It's sort of something that when you really think about it, it's like, it's amazing that, it's incredible that like everyone is operating this way right now.
Maybe to close up, if we broaden the scope a little bit, are there other problems that are in let's say biology, medicine, science, technology that you think are like worth working on and interesting and perhaps feasible, but people are not looking at as much as they should be? You know, maybe not even making a value judgment, but that you're curious about.
Aesthetically, I'm just really curious about like how to represent the molecular world in a way that people can understand and engage with. Like, I think it's just such a cool and beautiful thing. It's like a, you know, we were talking about kindergarten like worlds earlier. It's like this beautiful kindergarten like feels in my mind very colorful, even though it's like not literally sort of place to play around. And I think most people experience it as like flat triangles and squares in a biology textbook where it's like, you know, there's an arrow between like this triangle and this triangle. It's just like doesn't make any conceptual sense and is like confusing and annoying. And so I'm just like, I personally am really curious about like how to represent the molecular world in a way that is really compelling and feels super exciting.
From an education perspective,
I think it is artistically cool if it's educational, but I think it's more just because I think it's so beautiful. It's like if you can never seen a tree, it's like being able to see a tree. It's like that would be so great, right? You know, it's like this complex fractal thing and all this like light falling through the sort of tree branches. And I think like the molecular world is like that. It's just it's a kind of beauty that's not made accessible to most people. It's hard to conceptualize and you have to like do some amount of studying to sort of like build the world in your mind correctly.
I look forward to buying that art. Thanks, Laura.
Thanks for having me.
Find us on Twitter at NoPriorsPod. Subscribe to our YouTube channel if you want to see our faces. Follow the show on Apple Podcasts, Spotify, or wherever you listen. That way you get a new episode every week. And sign up for emails or find transcripts for every episode at no-priors.com.